Hello everyone, my name is Subishand Janasagran and my matrix number is 203034.
Hello everyone, my name is SACHINTHARAN, MANIMARAN and my matrix number is 202358.
So today, myself and Sachin we're going to talk about respiration.
Okay, so let's look at what is respiration. So respiration is a metabolic process where the living cell obtain energy by taking in oxygen and exhaling carbon dioxide from the oxidation of complex organic substance. So in this process, the energy ATP, which is known as adenosine triphosphate, shortly known as ATP, is produced by breakdown the glucose, which is further used by the cell to perform various functions. Next slide. Okay, so then let's look at aerobic respiration. So aerobic respiration is a cellular respiration that takes place in the presence of oxygen to produce energy. And the glucose will be break down into carbon dioxide, water, and energy in the aerobic respiration. Next slide. And the second type of respiration is anaerobic respiration, where the cellular respiration takes place in the absence of oxygen to produce energy. So anaerobic respiration can be divided into two, where the first one is alcoholic fermentation, where alcohol will be will be the product of the from this respiration, and then lactic acid fermentation, where lactic acid will be produced as the byproduct of this respiration. Next slide. Okay, so now let's compare between aerobic and anaerobic respiration. So the first one is for the aerobic respiration, oxygen is present, whereas for the anaerobic respiration, there's no oxygen. And then in the aerobic respiration, there is gases are exchanged in the form of respiration, whereas for anaerobic respiration, there is no gases exchange. And then the third one is in an aerobic respiration takes place in cytoplasm and mitochondria, whereas for anaerobic respiration, it only takes place in cytoplasm. And then the fourth one is in aerobic respiration, the glucose breaks down into carbon dioxide, water, and energy, whereas in anaerobic respiration, it breaks down into ethyl alcohol or lactic acid, then carbon dioxide and energy. Then finally, in aerobic respiration, all the higher organisms such as mammals have this type of respiration, whereas lower organisms such as bacteria and yeast use anaerobic respiration. But then in few cases, when there is a heavy activities, there will be presence of anaerobic respiration.
Okay, now I'm going to explain about the net energetics of cellular respiration. So let's look at there's four important cellular respiration. The first one is the glycolysis, grooming of pyruvic acid, Krebs cycle, and the electron transport chain. So if you look at the net of ATP that's being produced in glycolysis is 2 and in Krebs cycle is also 2. For the electron transport chain, 34 ATP is being produced. For the NADH, for glycolysis and grooming of pyruvic acid, 2 NADH is being produced respectively. For the Krebs cycle, 6 NADH is being produced. For the FADH2, for the only FADH2 is only being produced in Krebs cycle, which is the 2 net. So lastly, for the CO2, for the grooming of pyruvic acid, only 2 molecules is being produced. And for the Krebs cycle, 4 carbon dioxide molecule is being produced. So if you look at the net ATP that is being produced, is the total of 38 molecule is being produced in one glucose. So now let's look at what is Krebs cycle. So basically, Krebs cycle is also known as the citric acid cycle, is the main source of energy for cells and also an important part of aerobic respiration. It takes place in the matrix of the mitochondria for the eukaryotic cells and also cytosol for the prokaryotic cells. So the cycle harnesses the available chemical energy of acetyl coenzyme A into the reducing power of nicotine amide adenine dinucleotide, or known as NADH. So the glucose is oxidized to form pyruvate, which is then oxidized and enters the TCA cycle as acetyl-CoA. So this is the basic structure of the entire process of Krebs cycle. So let's look at the first step. So the first step is the condensation of acetyl-CoA with a 4-carbon compound oxaloacetate to form a 6-carbon citrate. Coenzyme A is being released. So the reaction is catalyzed by the citrate synthase. So for the second step, the citrate is converted to its isomer known as the isocitrate. So this enzyme, the enzyme that involved in this reaction is aconitase enzyme. So the thirdly, the isocitrate undergo dehydrogenation and decarboxylation to form 5-carbon alpha-ketoglutarate. A molecular form of carbon dioxide is released. So the isocitrate dehydrogenase catalyzes the reaction. So it is an NAD plus dependent enzyme. So the NAD plus is converted to NADH. So if you look at this diagram, you can see that the oxaloacetate combines with acetyl coenzyme to form citrate. So the citrate is further converted to isocitrate by using the enzyme aconitase. So the isocitrate is also further converted into alpha-ketoglutarate by using the enzyme isocitrate dehydrogenase. Here, the NAD plus is further converted to NADH. Next is step 4, where the alpha-ketoglutarate undergoes oxidative decarboxylation to form a 4-succinate-CoA, a 4C compound. The reaction is catalyzed by the alpha-ketoglutarate dehydrogenase enzyme complex. So one molecule of carbon dioxide is released and NAD plus is converted to NADH. So for step 5, the succinyl-CoA forms succinate. So the enzyme succinyl-CoA synthase catalyzes the reaction. So this is coupled with the substrate level phosphorylation of GDP to get GTP. So the GTP is transferred to its phosphate to where the ADP forms into ATP. So step 6 is about succinate is oxidized by the enzyme succinate dehydrogenase to form fumarate. In this process, FAD is converted to FADH2. So from alpha-ketoglutarate, it is converted to succinyl coenzyme by using the enzyme alpha-ketoglutarate dehydrogenase. So the succinyl coenzyme is further converted into succinate, where the GDP molecule is converted into GTP molecule. So the succinate is also further converted to fumarate, where FAD is converted to FADH2. So the in step 7, the fumarate gets converted to malate by addition of 1 water molecule. The enzyme that catalyzes the reaction is fumarase. So last step is that the malate is dehydrogenated to form oxaloacetate, which combines with another molecule of acetyl-CoA and starts a new cycle. So hydrogen is being removed and get transferred to NAD plus, forming NADH. So malate dehydrogenase catalyzes the reaction. So here you can see that the fumarate is converted into malate by using the fumarase enzyme, and the malate is further converted into oxaloacetate by using the malate dehydrogenase enzyme, where NAD plus is further converted to NADH. So now Sachin will explain about electron transport chain.
Okay. So the final stage of aerobic respiration is the electron transport chain, which is located on the inner mitochondrial membrane. So the ETC, shortly known as ETC, releases the energy stored within the reduced hydrogen carriers in order to synthesize ATP. So this electron transport chain, also known as oxidative phosphorylation, as the energy to synthesize ATP is derived from the oxidation of hydrogen carriers. Okay, so let's see what is ETC. Is ETC also known as oxidative phosphorylation? No, behind, not yet. Also known as oxidative phosphorylation, has three important steps, where the first one is the proton must be pumped to create an electrochemical gradient known as proton motive force. And then the ATP synthase use the subsequent diffusion of protons chemiosis to synthesis ATP, which is an important product from this process. And then the final step is the oxygen accepts the electron and proton to form water and then stops the production of ATP. So let's see, how does this work? The next slide. Okay, so the first step is by generating a protein motive force. So this happens when the hydrogen carriers such as NADH and FADH2 are oxidized and release high energy electrons and protons. So this happens in the Krebs cycle. Krebs cycle. Okay, then the electrons are transferred to the electron transport chain, ETC, which consists of several transmembrane carrier proteins. So as electron pass through the chain, they lose energy and it is used by the chain to pump proton H plus ions from the matrix. Then the accumulation of H plus ion within the intermembrane spaces, where you can see the blue color, the pink color from the picture, it creates an electrochemical gradient known as protein motive force. Next slide. Okay. Next slide. Step 2. Okay. So then the second step is ATP synthesis via chemiosmosis. So the protein motive force will cause this happens when the protein motive force cause an hydrogen ion to move down their electrochemical gradient and diffuse back into the matrix. Then this diffusion of proton is called as chemiosmosis, and it is facilitated by the transmembrane enzyme known as ATP synthase. So as the hydrogen ions are moved through the ATP synthase, they trigger the molecular rotation of the enzyme, and this will make the enzyme to synthesize ATP, known as energy, the energy. Okay, next slide. Okay, in this slide, we can see there is a reduction of oxygen, and this will eventually stops the ATP generation. Okay, how does this happen? This happens when the electron transport chain continue to happen, and it will de-energize the electron. The de-energized electron will be removed. So then the oxygen act as a final electron acceptor, where it will remove the de-energized electrons to prevent the chain from from blocking the block. So then the oxygen also binds with the free protein in the matrix to form water by removing matrix protons, and this will maintain the hydrogen gradient along the series. And then in the absence of oxygen, the hydrogen carriers cannot transfer energized electron to the chain, and ATP production is halted. So the total production of ATP through electron transport chain is just 34. And the overall production of ATP throughout this respiration, aerobic respiration, is 38. So next slide. Okay, so that's all from us. Thank you very much.